We have Dr. John Quinn from FlowJo, and the talk title is Post-Acquisition Quality Control in FlowJo.
Okay. Hello, all. All right, can you hear me?
Yep, you're all good.
Perfect. All right, so I am presenting here from virtual Hawaii, where if this was normal times, we would have all been perhaps at AAI right now. And I'm just seeing if I can just have Zoom on continuously for the whole weekend to pretend that I am in Hawaii as a little bit of a Zoom vacation. See if it works as well as going out on a real vacation. So the talk here is post-acquisition QC in FlowJo. I'm John Quinn, Director of Science and Product Development at BD Bioinformatics, aka FlowJo. My email address is john.flowjo@bd.com. And you can reach me there for any kind of questions, comments, etc. at any point. I have the QA portion of Zoom opened up. I will try to go through the whole talk and then circle back to any questions that were asked, but I will have that up. I'll keep that in mind, and feel free to type away and send in some questions. All right, I'm going to stop my lovely Hawaii video for the time being to give more bandwidth to what's going on here. And so the idea is I will spend about half the time with some slides, and then I will demo everything that I'm going to talk about in FlowJo, because I think it's always a good idea to show how to use the tools that I'm going to advocate for. Okay, so when I say quality control in FlowJo, what do I mean? Because it has to be post-acquisition, of course, and so there's a lot of quality control one might do in cytometry. What we do in FlowJo afterwards is to check for the consistency of your individual data files across the time of collection. And we can also look to try to normalize the consistency across different data points within a larger study. So the first one is important because I think most everyone who's done some flow at some point in their life has spotted some interesting side population or spur population that they've tried to hunt down for why is this different, which markers are brighter or duller, and found that in the end, it was just something that was based off a little blip in the acquisition due to a difference across time. That either the cytometer hadn't yet settled down to laminar flow, or there was a clog or something of the like that caused some small portion of the data to just universally look brighter or dimmer or different in some way. And so checking time right off the bat on your individual data files is a great way to make sure that the differences that you're seeing in your data are due to biological phenomena as opposed to just some kind of just instrument or collection artifact. And so we have tools like a built-in QC indicator in FlowJo, and then plugins like FlowAI and FlowClean that are designed to help you pick out those artifacts before you even start analyzing the data and go down any rabbit holes trying to chase down what an odd or side population might be that turns out to be just an artifact. The other tool that I've mentioned, CytoNorm here, which we're going to use for across time, is the idea that if you want to compare files that were collected months apart, weeks apart, on different cytometers and so forth, that we generally look at fluorescent parameters in terms of intensity, which is a dimensionless parameter, and it's not necessarily true that 100 intensity units in one data file equals 100 intensity units in another file. These differences in brightness could, again, just be attributed to differences in a machine over time, or even just different cytometers running that have different setups. And so we'd like to make sure, again, that when we're doing comparison, that the differences are due to some kind of biological phenomena and not just machine setup. So let's talk about those tools. So first thing I should do, as I've just mentioned, 3 plugins, is to mention what a plugin is. Forgive me, I will probably use at least a little bit of jargon in this talk that I won't have time to spend a lot of time explaining. Please write me with any questions. And also check out the Learn tab, which I believe is right there visible on the screen, which has a number of webinars, questions, help, etc. For most of these topics. But a super brief intro to plugins are that they're just extra programs that can be plugged into FlowJo to give you a little bit of added functionality. They're all free. They're available on the FlowJo Exchange, which I've highlighted in the screen. Is just off of www.flowjo.com. The Downloads tab, scroll to the bottom, and it'll say FlowJo Exchange plugins. And then you'll be able to go shopping for whatever extra tools that you want. There are about 40 up there now. And as I'm going to switch to the next slide, you can sort by tools for either FlowJo or Seeky or both. And then based on whether you'd like tools that do quality control, dimensionality reduction, utilities, clustering, visualization, or scripts. And so this is just a screenshot where I've turned on quality control and tools for FlowJo, since that is the topic of the day. And what we'd like to show you here is that you've got FlowAI, FlowClean, and CytoNorm as options that you can use within FlowJo. FlowAI and FlowClean are, again, both tools that will look at your data across time. CytoNorm is one that will normalize your individual files across the time of collection. CytoNorm is one that will look at experiments across time and try to normalize out batch effects. All of these are published methods, published peer-reviewed methods that were written originally as R programs and vetted by our peers and are freely available to use via R if you'd like to. But the way we've set it up in FlowJo is that you can, from within FlowJo, just run these. The R package will happen in the background, do its thing, and immediately just import the results into FlowJo. Some will produce results, some will produce extra files, which will appear as derivatives on your computer that you can make use of. And so it's all tools that are generally published, reviewed, freely available in a number of places, and we try to just make it easier using them within FlowJo. Okay, so let's start with each of the tools then. So first, I'd mentioned that there is a built-in QC indicator in FlowJo. So if you open, this is version 10, and look at the cytometry band, there is just a check sample quality button. So the arrow is pointing at it. And if you click that next to each one of your files, you'll see there's a circle, and you can mouse over it and it'll give you text that tells you what it means. But generally, the circle is going to color somewhere between green for good and red for bad. So the gradation of colors is green, blue, yellow, pink, red in descending order of quality. And so all this circle is meant to do is catch your attention and say, perhaps this is a file that you should look at versus time, because perhaps there is some kind of artifact you'd like to gate out. And so anything that goes yellow, pink, or especially red, you would like to double click on the file perhaps and check out versus time. The other thing you can do is right click on any file. So in this example, I've right clicked there on file 5, which is a pretty good sample. It came out blue. And I've chosen to inspect it. So inspect, just like iTunes, just tells you everything there is about the file. And so what this is showing is a graph of every parameter in that file, where time is the x-axis in all cases, and the y-axis is the median of each parameter for a bin of about 1/10 of a second. And so what we expect to see, since the cells should not arrange themselves in any way in the tube as they come through the cytometer, is that over time you'll see some gentle oscillation above and below the median, which is the dashed line down the middle, as a group of larger cells or cells with greater binding affinity or lesser binding affinity to a particular parameter pass through, but it should generally even out over time. So again, gentle oscillation around the median. The dashed line on the top and below are showing a standard deviation, which is auto-scaling, as you'll notice down here at per CP, which is looking a little jagged, so perhaps the blue color as opposed to the green. You'll also see that a standard deviation is all the way up to the top of that plot. So it's giving you an indicator of, did things pass through evenly? So you might see pink or yellow or red if perhaps there was a startup point where at the very beginning of acquisition, you don't have laminar flow, or maybe you're using a plate and there's a moment where the sample's been disturbed by the needle being added, or aspiration or something to that effect. A clog where there's no cells passing through for some amount of time, or fewer cells passing through, or just a general trend where the data is just getting brighter or getting dimmer or something to that effect. We've got a non-zero signal across time. Those are all things that will flag one of the colors. And so this is okay. It gives you an idea in just a snap of the finger or two, based on glancing at the colors, of how good the overall experiment is, if there's a few files you need to look at, but it is largely heuristic. And so I really like using some of the algorithms to do this kind of sample quality analysis. So FlowAI, actually my favorite of the two, will check all of the flow rate, your signal stability, and the dynamic range by running this algorithm, which was written by Gianni Monaco from the A-Star group. If you go get the algorithm on the FlowJo Exchange, there is also a link if you'd like to read the citation, which is just what I've just popped up on the screen here on this slide. So what FlowAI will do is it will plot your data in 1/10 of a second increments for how many cells have passed through in that 1/10 of a second. And you can see in this plot here that everywhere that the rate went out of either too high or too low compared to average, we draw the little green circle and say this is 1/10 of a second that we're going to remove out of the data. So this is a pretty nice file where there's a little bit of noise at the beginning. It settles down to an acceptable amount of oscillation. We get 2 points down there towards the end that get a little low for choosing average, and then we've got the stopping condition there. It'll then also look at signal stability, and it'll check through your data to make sure that every one of the parameters you feed it is about equal over time. We're always going to assume that there is some variability just from different cells expressing different amounts of any particular antibody that you've bound to it. And so this is just looking for long, consistent shifts. And if it finds a place where the signal just jumps up by a modifiable threshold, then it will call that a change point, and it'll remove the data either before or after that change point, whichever way you are able to keep more of the data. And so we can see in this data right at the end, basically as we were turning off the cytometer, there's a change point where the signal goes to 0, and that's listed as a change point that is taking out perhaps the last, whatever, 1/2 of a second or something like that from the acquisition. And then the last thing it'll do is just check to see if there are any cells in either the lowest or the highest bin, the bins where it is likely that most of the cells present were actually outside of the measurable range of the cytometer. They just get lumped into one of those bins. And then we just give you a chart at the end that tells you how many cells were taken out due to either one of those conditions being met. So here there was 85 cells that were the lowest bin of FL1 and 73 on FL3, et cetera. And we'll take those out too. And what gets left is just a gate called good events. And you start your work from the good events portion. Data quality checked, at least for that sample across time. And I'll show you in FlowJo that it's a groupable action, so you can apply it to all your data just by drag and drop. So that's FlowAI. FlowClean works in a fairly similar manner in that it is again going to look for consistency in your data. FlowClean is going to do this by basically auto-partitioning your data, just looking for saddle points and so forth, and saying for each cell, what type of cell is it based on? Was it high, medium, low for all the different parameters, calling it a population of some kind without trying to assign any biological meaning to it, just saying it was high for this, low for that, etc. And then seeing if there are any populations that are distributed something besides evenly across time. If they are all in the first couple of seconds, then that's an indication that particularly high for all the parameters or something like that might be an artifact produced by the machine. And then it'll take out those cells just like FlowAI took out the ones that either missed by flow rate, etc. So similar tools there. I tend to like FlowAI mostly because I feel like I've got some better control over it through the settings and the tunables, and it makes a really nice report. And it's, I laugh a little bit, but it's important to be able to know why an algorithm has stripped out the cells, and I love the reporting functionality out of it. So when I get to demo time, I will demo that one for you. Okay, the last one I want to talk about was cytonorm. And so this is again meant to address comparing different files that were collected at either different time points, different cytometers, or something like that. And so the general idea is that if you've got a comparable control at each time point, you can use that as a tool to normalize the rest of your data to. And this is actually a tool written by Sophie van Gassen, who is the creator of FlowSOM, SOM being a self-organizing map, FlowSOM being a clustering algorithm based on self-organizing maps. And for more discussion on FlowSOM, we have a number of webinars, and the plugin itself contains both a link to the paper and some general discussion of what it is, but I'll leave FlowSOM at that for now. So in any case, you will be unsurprised to find out that the cytonorm algorithm takes advantage of a self-organizing map that is going to be used to identify the structure of the data and then align it. So the general gist of this is that if we assume we've got a control sample in every one of the different time points that we might want to look at, we can use FlowSOM to figure out the populations within the data. We can figure out that there should be a certain number of cells that have a particular profile in them, or it doesn't have to be a certain number, but just cells that have a particular profile in them, and we can figure out how they're distributed. We can then assume that in the samples that we are going to use those controls for, that they should have an equivalent distribution, that wherever the negative peak is should pretty much line up and have the same, the same spread as the representative control for that. So as long as we have a control for each of the time points, we can use a spline as a way to do some linear interpolation and align the data so that negatives all end up at the same place, and we can actually compare the relative positivity of the rest of the peaks in it based on having aligned everything else in the data based on the distributions of the various control samples. And so that's the plan for cytonorm. Okay, so having used half my time to tell you about the theories behind them, let's crack open FlowJo and I'll demo how to use them. Okay, ending PowerPoint. All right, so first off, I have an aptly named bad sample and good sample loaded into FlowJo version 10. I have moved my cytometry band under the FlowJo tab, and I'm going to click check sample quality, and we'll see that we get a pink circle under bad sample and a blue circle under good sample. If we just mouse over just to the right of these circles, it'll tell us for blue, sample quality was okay, and for pink, sample quality was irregular. We could, at this point, do some further inspections. One thing we could do, we could right-click on one of these and hit inspect, and then just check out the medians across time here. If we want to see if there was anything interesting, using the other arrows, we can move back and forth. The bad sample looks, as expected, a little sketchier. Okay, and then if I wanted, I could draw gates on these just manually by double-clicking, bringing up time on an axis, and perhaps trying to gate out any portions that look bad. So check sample quality, indicator, heuristic gates, if you'd like to do so. To use FlowAI on these, we can go to our workspace, plugins, pick FlowAI. Of note, you have to have picked a sample for it to run on. If you haven't picked anything, it'll just all be grayed out. When you run a plugin, it'll generally say, you have to save your workspace so we have somewhere we can write the results. So I'll say yes. We'll call this MetroFlow. Okay, MetroFlow workspace. So when FlowAI starts up, it'll ask you which parameters you would like to feed to the algorithm. I'm just going to pick them all. You can use either the uncomped or the comped. The uncomped are probably better in that just in case there is some artifact of compensation, it won't factor into your sample quality checks. But since we're just looking to deal with consistency, probably either are fine. You can choose which of the checks you'd like to run. So I showed you that there's a flow rate, signal acquisition, and a dynamic range check that you can do. You can pick one, a combination of two, or all. I usually leave all on. You can set the sensitivity for the flow rate, how many seconds you want to break the bins into, what the statistical significance you'd like to use is. You can use these settings to control the change point penalties for looking at signal stability. One thing to note is that the authors of the FlowAI algorithm have suggested making this 500 after initially suggesting 200. The bigger the change point penalty is, basically means the bigger the change has to be counted as a change point and to take out some cells. So by making it bigger, you actually make it less sensitive. And so I think a bigger number is nicer there. 500 is the recommended default now. You can choose to change the dynamic range, to check the dynamic range either on the upper or lower bound or both. And I like turning on remove outliers, which will, if you look at signal stability and you've got just one blip where you get a spike in signal, it doesn't have to note it as a change point. You could call it an outlier without having to remove a particular large part of the data. I also generally like saving the R script and output messages if I'm trying to get this started, if I've just installed it, in that it'll run. And if anything happens, everything that happened in R gets recorded. You could just read the text and see if there was an error and maybe how to fix it. And if that sounds tricky or complicated, it's what you can call our tech support, FlowJo at bd.com. and show them that, and they should be able to diagnose your issue near instantaneously. So I generally turn that on. So I'm going to say OK to this. Okay, we see a folder has popped up on my desktop called MetroFlow. If I open that folder, we'll see it's created a subfolder called FlowAI as it's running this script. It's starting to generate some files. It's created a good events and a bad events gate. So if we, okay. Oh, so there's a question on will these plugins work with FCS files from spectral flow cytometers like Cytek? Yes, all you need is time. as long as you've got time, we can look for consistency in time so that, we can make sure it's not an artifact. Okay, I'm gonna just drag in the bad events and the good events. It's already on time. You can see where there's, so very few cells were taken out. Looks like it's a couple of bins here that we can see stretch vertically across time. And then if we want to know why were those 3 or 4 tenth of a second bins taken out, we can go look in the output folder for good sample, double click on the web page, which has all the information, check out the flow rate for that particular sample and see that just the start here had a little issue. It's the same picture that I used when making my slides, which look fairly familiar. And a couple spots there that the signal stability was pretty good and that there was a handful of cells pulled out for being in the upper or lower bound of the dynamic range. And so that's why it gated out visibly 1, 2, probably looks like that's 3, 4. We actually just appeared to be 1 in the picture because they were so close together, why it took out these handful of events. So that was what FlowAI looks like on a good sample. And then it's always nice to see what it looks like on a bad sample. You can either drag the FlowAI icon to the entire group to run it on all the samples in a group, or either just to another sample if perhaps it's a demo and you've only got one other sample loaded into this workspace. Okay, so that's going to run in a second. And we're going to see that there is now bad sample and its information filling in that folder. If we look at the bad sample figure, and it's going to show that it took out, if we look over here in FlowJo, it's going to show that we took out something like 83% of the events. And if we glance here at the flow rate, we see it was just a disaster here. So this is what happens when your flow rate varies dramatically, maybe stabilizes down here, jumps back up, maybe stabilizes at the end. So the algorithm is trying to figure out whether the higher rate or the lower rate are actually normal or average. The lower rate had this long period at the end that makes it win on what seems to be the most average. So everything at the higher rates is taken out, that it's just dramatically variable, is a bad sign. The signal acquisition actually looks okay, but then at the end, we see that there, especially in that highly variable part, there's tons of cells that get smashed up to the upper or lower range and get taken out for dynamic range purposes. So anyway, that's what happens when you run a fairly bad sample through FlowAI. All right, I'm gonna move right into CytoNorm. We have about 4 minutes here or so let's just do it. Okay, so there's actually a second type of plugin. If I go to plugins, when I start this up, I see here's my list of all my plugins, but there's also the ability to add a workspace plugin. So there are some plugins that work on an entire workspace, and one of those is CytoNorm. And oops, I have to start a new one because I already ran it in this workspace. So we'll just start a new workspace, go to plugins, add a plugin, pick CytoNorm, and it's going to ask for me to point to a folder that I would like to run CytoNorm on. So for my desktop, I have this CytoNorm demo folder that I'm going to point to, and it's got a number of files in it, and I'm going to pick out these files. It'll pop open an interface where it'll say, which are your controls, and relate them so I know which batch they go along with. And so if I've got a couple of different time points here, year 0 and year 4, I can say the controls are maybe year 1. We're going to expect that we should see that those are maybe a negative for each one of these files or a baseline, and that they go together with the same patient at the next time point. So I'm going to say these should go together, these should go together, and these should go together. I would like to include the controls in my normalization because I'd like to see them normalized to compare it to the rest of my data and say, okay. So in this folder, CytoNorm is going to start running. It's starting to create some R files here, but we've got 2 minutes left. So I did this cooking show style. I ran this just before the start of the meeting and I have the results here. I think this took 5-6 minutes to run. But anyway, let's drag in the original data and let's drag in the normalized versions of the data to show what CytoNorm wants to do. Okay, and just for cleanup purposes, we'll go grab, oops, zoom controls in front of my gates. We'll go grab, say, a population of lymphocytes, or close enough, and go to the layout editor and say, all right, if I take my lymphs and I show something like one of these fluorescent parameters as a histogram, I can overlay all of the rest of the time points. And maybe I'll right click on it. Oops, sorry, I duplicated it. I'll right click and half offset them. Okay, so you can see that we've got a few differences. So I've got them every other here. That where's the background? In each of the year 1 files, it's a little bit lower than each in the year 4 files. And so that makes us suspicious that we can directly compare where positive might be. In the normalized files, so we can drag this in, switch to CD3, might not have had a tremendously good cleanup gate there, but I think this will still be okay. Histogram.
Just to let you know, John, we have about 5 minutes left.
Sounds good. This is the last thing we're showing, just an overlay here of each of these. And we will stack them up in a half offset. And just to show that now the algorithm has normalized all the negative populations to be at about the same spot, and the positive populations are normalized as it works out. You know, not all of them will have a very big positive population, and that's part of what the algorithm will do, but where there is one, it then lines up. And so it can take out something that's just, oh, it looks like this entire distribution sort of matches what that distribution has, and it's just more condensed, and normalizes for you. Okay, so that was the end of what I wanted to demo. I see there's at least one question here in the chat. Let's see, and it's why did you fill 1, 2, 3 to apply cytonorm? Okay, sorry, yeah, I went through that fairly quick. And so what I had there when I started up cytonorm was 3 different patients. And I had for each patient a control file at an initial time point. And I wanted to make sure I matched the right patient with his ladder file that I wanted to normalize against his control. So 3 patients, I put 1s in for each of patient 1's files, 2s for all of patient 2's files, and 3s in for all of patient 3's files. Good question. Okay, and going to the QA box, looks like folks used the chat instead. So I think we are clear there. And I think we've got 4 minutes if anybody wants to ask a question now, or else we'll turn it over to Tina to talk some MetroFlow logistics.
John, I actually have 1 or 2 questions myself, so I might ask those if we have. So when you go to the Flow AI plugin, you know, you have all of the options for the different parameters for signal acquisition check, right?
Yep.
I was curious, you know, you have all the parameters selected, and you said that, you know, the uncompensated is better. So would you ever recommend not selecting all fluorescent parameters and just using 1 off of each laser line, or do you always select all? And then as a follow-up to that, does it make any differences in the amount of time it takes to run based on how many parameters that you check?
Okay, so good questions. So there will be some amount of difference in time on how long it takes to run, but it's probably not appreciable. The algorithm itself runs fairly quickly. It runs something like 20 seconds on 500,000 events, 8 to 10 parameters. So, you know, maybe if your variability is 19 seconds or 20 seconds, it's probably not something you perceive. So no worries about that. If it's all the same to you, just run them all. I tend to just run them all because I want to see if any of the parameters had any kind of nuance in them. You may certainly choose not to run some if you decide for some reason that there's a parameter that you maybe you don't want it to check, because if it finds a time-based change point and takes out some cells, it'll take them out for that entire time point, so all of the cells. So what if, simple example, you ran a kinetics experiment and there is some parameter that you are actually measuring a change in time? Definitely, absolutely leave that out. But generally, if all the data, you just want to make sure they're consistent, I would just select them all.
Okay, that sounds good. And I have one more question for you. So when you're talking about that change point penalty, I know the recommendation by the authors and by you is to kind of increase it to 500. So is there any way that we can change it so that the default setting there is 500 as opposed to having to manually change it each time?
It's a good request. I think, unfortunately, because it's a plugin as opposed to something that we have preferences for in FlowJo, that's going to be on us. So we should actually take care of that and do that. So sorry on that one. We'll get to that.
No, it's okay. I was just curious to see if there's a way of doing that I was not aware of. So that's good to know. And I think that's definitely all the questions that we have, and you've been very wonderful at answering all of them. So thank you so much, John, for, as usual, for a wonderful talk and presentation of FlowJo.
Oh, well, thank you all. I appreciate everybody attending. Have a great weekend. Take care.